So thank you everyone for joining us this morning. My name is David Fleck. I'm founder and one of the managing partners of FreeFlow. Joined this morning by my partner, Kevin Barrett, who's also a managing partner, and by Dalton, who is the founder and CEO of Miist Therapeutics. Super interesting company. Love what they're doing. Excited for everyone to hear more about it. The company they basically have had this realization that through using fit, so it's a Berkeley-based company. As you may recall, we invest in companies coming out of specific ecosystems, and we've developed, an ecosystem-centric approach focused on Caltech initially, and then we 2 years ago started to invest in the Berkeley ecosystem, and I found that to be a very promising ecosystem. I live in Berkeley, so I see it firsthand. But more importantly, what Miist is doing is they've developed a novel inhaler that's clinically validated to enable existing previously approved medications to be fully absorbed and take effect generally in less than a minute, perhaps 30 seconds, which is about 100x faster than it takes for many pills to take effect. And this really has profound implications for how one might think about various types of conditions, including migraines, which Dalton's going to talk about today. And then there's other potential applications as well, where there's really the need for speed in terms of just the efficacy of various treatments. So with that, let me pass it over to Dalton to provide some introductory context and remarks, and then we'll rejoin. I'll turn my camera off, Dalton, just to give you the stage. But I encourage everyone in the audience to put questions in the chat. We'll get to all those questions along the way. But with that, let me turn it over to Dalton. Again, thanks for joining us this morning.
Awesome. Well, thank you very much, David. And yeah, excited to be here and be talking with everybody, sharing a bit about our story. So I'm thinking that the best way to do this here is that I'll start off with, I like to start at the thesis layer in terms of what we think is important here and what we're doing. And then I'll go into a bit of the backstory as to how we actually developed that thesis and started working on this company, and then provide some details on the lead asset that we're developing right now. So let me go ahead and share my screen here while we do this. And David, just let me know when you can see this so that I know everything's working.
Looks good.
Great. Okay. All right. So starting at the thesis layer, so building off of what David was saying, since day one of the company, we've had the same general idea, which is that speed is a really powerful and untapped efficacy lever in medicine. And essentially, if you're able to take a drug, make it work significantly faster, you can actually dramatically increase the efficacy of that drug, so much so to the point where if you can get it fast enough, you can drive it all the way to best-in-class status, meaning it's the most effective drug in a given field. And so for us, this was really interesting because typically in bio, you see companies that are either modulating chemistry or biology, and they're developing new drugs to try to create more effective drugs. And our founding team didn't have a chemistry or a molecular bio background. We were sort of an engineering and physics background. And so we found this other lever and we said, hey, look, this is actually, squarely within our disciplines and our area of expertise, and we can use it to compete in the biofield and create this new category of best-in-class therapies. And so sort of was this interesting lever that we found that we thought we could pull on to create better medicines. The backstory as to how we got there goes back to my time when I was doing my undergraduate studies, and I was actually exposed to tobacco addiction for the first real time. That's sort of where everything started here. And around the same time, what I had essentially was that I had exposure to probably 20 to 30% of students on my campus at Villanova who were heavily addicted to e-cigarettes. I had a younger sister who was in middle school who was saying that all of her friends were starting to use e-cigarettes, a grandmother who was a lifetime smoker diagnosed with COPD, and all this stuff just kind of hit at once. And it essentially showed me the spectrum of tobacco addiction going from your youngest kids to your oldest adults and just how big of a problem that was. And it got me really interested in this space. So I started researching the tobacco addiction space, and one of the hallmarks of that field is how ineffective all the treatments are. There's several FDA-approved treatments for smoking cessation to help people quit, and the failure rates famously are over 90% in that space. And as I was looking into that, there seemed to be a really logical reason for that, which was a clear timing mismatch. And so what's happening there is when somebody quits smoking, obviously they get these episodes of intense cravings and withdrawals. And you have a really short treatment window to actually treat those symptoms before somebody's going to relapse. And typically you're looking at about 5 to 11 minutes. You need to treat within that time period. And when you look at the therapies that are approved by the FDA, the fastest one works in about 30 minutes. It reaches its peak therapeutic effect in 30 minutes. So it's just clearly too slow to actually work in the therapeutic window that patients need. And that was a really interesting realization for me because it seemed so obvious that there was this mismatch. And so that was the first nugget for this idea. And then I looked to see if that existed in other areas of medicine, and it was very clear that it does. And there's actually really strong clinical data in other indications that shows speed is a very potent efficacy lever for medication. And typically it tends to be centered around CNS conditions, acute CNS conditions. And so once we came across that realization, we convinced ourselves that you don't have to discover or develop an entirely new drug to create a more effective therapy. You could actually apply the speed principle to make existing drugs much more effective. And so we centered this company around that. And after we had that, the idea was, okay, now how do you actually accomplish that? What technology do you need? And so I have some slides to get a little bit more into that. And I'm going to be focusing on migraine, which is our lead asset that we're focused on at Miist right now. And so I'm going to walk through a little bit of the underlying neurobiology in migraine that makes it an interesting use case for us, why speed matters, where our technology fits in, and sort of use that to frame the company and what we're building here. So let's start with one of the really interesting findings that served as some of the early inspiration for our work here. So this is a very high-level overview of a trial that was done by Dr. Rami Burstein at Harvard Medical School in migraine patients. And the purpose of this was to show the relative efficacy of the same exact migraine drug if you treat early in a migraine attack versus late in a migraine attack. And what he showed in his study was that the timing of treatment can cause a 6x difference in efficacy. So for patients who were treated late in their migraine attack, after the migraine had sensitized the central nervous system, efficacy was about 15% for those patients. And all of the patients who actually received migraine relief relapsed and had another migraine within that same day. So a very poor outcome. They then took same drug, same dose, delivered it to patients very early in their migraine attack before there was any sensitization of the CNS system. And on the next slide, we'll get into exactly what that means. And they saw efficacy jump up to 93% complete migraine relief, and 0 of those patients had a migraine relapse within that same day. And so it was just a very stark difference in the efficacy profiles here. And this data got us really, really interested and bullish on migraine as one of our pipeline therapies. And, we've been working on that for over a year now with some really good results. So let's go a little bit into the science of it here, because I think that this is really important to understand. Because what we're doing here, it isn't applicable for every treatment or every condition in the world. You wouldn't use our approach, our speed approach, if you're looking to treat something that is a very steady state condition. For example, something like high cholesterol. There's no value of speed in treating a condition like that. But in a condition like migraine, because of the neurobiology aspect that we can talk about right now, it's a highly potent efficacy lever. And so you can think about what we're about to go through here, and then you can put it onto other areas that are acute CNS conditions, where what you're looking at is actually a peripheral brain target, essentially. So to sort of highlight that, migraines have stages, and each stage is associated with a different amount of efficacy, essentially. And so when you first have a migraine, what happens is that it's starting in the peripheral nociceptors. So it's outside of the central nervous system. And so in your first 10 minutes of a migraine pain, you have this isolated, relatively weak pain signal that exists in those peripheral nociceptors. And that's the site that these migraine drugs bind to. So if you can deliver a therapeutic dose of the drug within that period of time, it binds to those receptors, it effectively shuts them down, and you see really high potency. And that's what we saw in the Burstein study with the 93% efficacy for early treatment. Then something really interesting happens over the next 10 minutes to an hour of a migraine, which is that the pain signal coming from your peripheral nociceptors is basically just barraging the second order neurons within your central nervous system. And eventually, as that happens, it starts to sensitize those CNS neurons. And what I mean by sensitize there is it basically makes them hyperactive. It sort of trains them to overreact to stimulus, and even to become autonomous and to create their own pain signals. And so the big shift that we see in this 10 to 60 minutes is increasing sensitization of these CNS neurons. And then after an hour, they're actually able to create and amplify their own pain signal. And that is a really critical point, because once that happens, you lose the ability to effectively treat a migraine. Our migraine drugs, all the migraine drugs that are out there today, are peripherally acting. So they're not crossing the blood-brain barrier, getting into the central nervous system and shutting down those neurons. So you have to catch it before the pain migrates there. And so that's why we see once that happens and you try to treat late in a migraine attack, the efficacy drops dramatically, down to about 15% in that study. So this is the race that we're running, basically, is we're trying to beat this central sensitization phenomenon that happens in migraine. And the big problem with pills is that they just simply can't win that race. Migraine pills specifically, they take about, on average, 2.5 hours. Some take all the way to 4 hours to be fully absorbed in the body and take effect. And so by the time that they've reached their peak effect, you have a fully sensitized central nervous system that is no longer responsive to medication, and you get very poor outcomes. So that's exactly what's going on in migraine, but a very similar thing happens in lots of other acute CNS conditions. This peripheral to central sensitization phenomenon almost perfectly maps to lots of different areas of acute pain as well, as an example. So things like migraine, acute pain, anxiety and panic attacks, insomnia, withdrawals, they're all mediated by a similar effect here. And these are the areas where we can have a really big impact with our treatments. So just want to highlight some of the early data that we've generated so far. So after we realized that, exactly what the company was going to look like, and we started developing the technology, we knew that the most critical thing for us to do as early as possible was going to be to validate our thesis and our technology in humans. And so we spent the first 2 years of the company really in the lab developing our inhaler technology, which I can show everybody here. So this is the inhaler that we have generated here. This is what created the data that you see up on the screen here. And this is what we call our microparticle inhaler. And we'll get into a little bit of that sort of naming and why we call it that. But once we developed this technology and we actually had it in this handheld format, first thing we did is we took it into a clinical trial with 10 patients using a well-known small molecule. And what we were looking at here is how fast does our treatment actually work? And so this is a PK graph. We did an arterial PK study. And what you can see here is that these peaks on the left-hand side are for 3 different doses that we tested. All of them reached peak plasma levels in the bloodstream 30 seconds after dosing. And this validated that our technology essentially works as good as it possibly could. So this 30-second Tmax here is on par with an intravenous injection. And it's, as David said, a few hundred times faster than what you typically see with tablet or oral medications. And there's a useful comparison that we can go over here. So this graph here on the left, this shows relative absorption times of different treatment modalities. So, we're at the top of that list there with a 30-second onset. So we're similar to an IV injection. An IV injection you can only do in a clinic, so it's not really much of a product per se. And so the next best product that somebody could actually use in a home setting would be a subcutaneous injection. So that typically gets drugged into the body in about 15 minutes, which is, the best currently approved method. But obviously there's lots of needle aversion as well, and people aren't big fans of injecting themselves. So if you don't want to inject yourself, then your current best option is a nasal spray. There's some variants on nasal sprays, but typically you're looking about a 30-second Tmax. With inhalers, normal inhalers, not microparticle inhalers, you're looking at about an hour for absorption. And then with pills, you're looking at about 2.5 hours. And so as you can see from this here, there really isn't anything that comes close to the speed of our microparticle inhaler. And with a subcutaneous injection specifically, we just don't see people walking around injecting themselves all the time unless you absolutely have to for something like insulin if you're a diabetic typically. And so I think that what we have here is a really unique opportunity where everybody wants their medicine to work as fast as possible. Everybody wants their medicine to be as effective as possible. We can actually deliver sort of both of those in the same product. And we can do it in a way with this inhaler that actually allows for productization at a large level, right? You can carry this inhaler around in your pocket, in your purse throughout your day. And then if you have a migraine or you experience pain or whatever the condition may be, that's something that you can just sort of pull out and use on an ad hoc basis. And so the general idea here is that we can sort of introduce this new category of instant acting medicine to the world in a way that's actually manageable for them to use and integrate into their life on a daily basis. The other thing that I want to touch on here is sort of the model that we use as a company, the development model, because we look very different than a traditional bio company. Traditional bio, you have a 10-ish year time horizon to go from discovering or designing a new molecule through your litany of preclinical testing, clinical testing, FDA approval. It's a very lengthy process, of course. And one of the big benefits to our approach is that you can largely condense that down. And so generally speaking, our programs tend to be about 3 times faster and within the range of 20 to 40 times cheaper than what you typically would spend on the development of a best-in-class therapy. And the real reason for that is because we aren't developing new molecules. We're taking existing molecules, making them work significantly faster, and using that speed as the efficacy lever to achieve best-in-class status. And when we do that, we get to use all of the existing safety and efficacy data that exists for that drug. So it really sort of condenses and shrinks the amount of new data that we have to generate. Essentially, what we need to show for our therapies are the route specific changes. So if we use migraine as the example here, sumatriptan, the drug that we use, is approved in different dosage forms, being used by millions of patients. All that we need to show on the safety front isn't that the drug is safe for people to use. That's well established. It's just that when you deliver it to the lung, there's no lung specific problems. And so our safety program is a fraction of what you would typically see in new drug development. And then on the efficacy perspective, we aren't taking any mechanistic risk of, oh, is this drug actually going to be effective in treating migraines? That's an established thing. We are just sort of working off our thesis that by getting the drug into the body, 250 times faster in this case, we see a significant boost in efficacy. And so we can very easily compare that in a trial against the sumatriptan pills that are out there, show that efficacy increment, and then use that for our FDA approval. And so we can typically go from concept to approval in about 3 years, going through these stages here, which are your initial lab validation and lung study in animals, and then going through a single PK study in humans in year 2, and then your pivotal trial after that. And so that's a really nice benefit to these programs as well, is that everybody's trying to get to the same point. They're all trying to get to best-in-class status, and we just aren't competing on the same fronts as everybody else to get there. We're using the speed mechanism that really nobody else seems to be trying to play into. And so that's I think a good overview of what we're working on here as a company. And so, David, I'll hand it back over to you and open it up. And specifically, I didn't go into too much detail on the actual function of the technology and talking about our microparticles in anticipation that there might be a question about that you want to ask. So we can cover that however you'd like.
Sounds good. Thank you for that. And feel free to take down that share.
Yeah.
First question that I have and always like to start out with is the origin story of why you do what you do and why you chose to build this company, because there's usually some pretty strong energy around that, and just the reason why the founders we work with who are doing really hard science, it's not usually because of a spreadsheet. It's usually because something pulled them in along the way. So yeah, just was curious if you could just explain a bit about why you started to do this, investigate this, and then eventually dedicate your life to it.
Yeah, absolutely. So, it started in the tobacco addiction world with sort of the experience that I referenced when I was younger, and seeing all of my classmates and my younger sister and my grandma and everybody being exposed to those products. That was my foray into this space, and that's where I first discovered that there was this relationship between speed and efficacy in medicine, and lots of conditions had these very short time windows where you had to treat within that window. So that was sort of the birthplace of the idea. And at that point in time, the first concept we had was specific to smoking cessation. It was that we've clearly identified you need a rapid therapy. If we can develop a treatment that works fast enough to meet that time window, we could actually help people quit smoking. And the majority of people do want to quit. You have about 55% of smokers that are trying to quit every single year. It's just incredibly hard to do so. And there really are no pharmacologic agents to help them right now because they're all too slow. And so that was the world that we started out in. And that was largely based off of just my experience with my family members and my classmates, and really thinking that we could make a difference there. And once we generated our original clinical data, that sort of served as a jumping off point for us to really say, okay, we've now validated our thesis. Because not only did we show that the technology worked in 30 seconds, but we also showed in that study that we had efficacy levels of 93%. So that was a smoking cessation focused study. And we were looking to treat patients' withdrawal symptoms and cravings who were smokers. And what we saw in that was we reached peak plasma levels within 30 seconds, and then we achieved a 93% reduction in their cravings and withdrawals within 2 minutes after they actually used the product. And so for us, that was thesis validation. That was, here's this incredibly rapid onset, and here's this very rapid pharmacodynamic effect that follows. And so here's the sort of speed equals efficacy shown with our own product in patients. And that was a platform level validation for us. So we then said, okay, what do we want to add to our pipeline, right? We don't need to focus and be a single asset company here. There's a lot of things that we can do. And so we selected migraine as the next best place to start because, some of the data that I showed, there's, already a very validated relationship for speed there. And then also my wife gets terrible migraines. And so she gets 5 to 7 migraines every month. And we've tried all of the different pills that are out there, and none of them, really work. So, I suppose there's a pretty big personal tie to that as well, and really having a direct line of sight into what matters to a patient, what they want, being able to have her and my mother-in-law, who also gets very bad migraines, as sort of early patients to bounce ideas off of and build things was helpful. So yeah, a little bit of a personal tie for the problems that we're working on, I suppose. And it's always nice to have the wife invested in our therapies, because I don't get in trouble for staying at the lab late. I just say, it's all right, I'm solving your big problem.
That's funny. Um, and then as part of it too, just another question that drew us in really early on was one word, which is physics,
Yeah.
and just how you can use physics, which is just so interesting. I mean, obviously, physics is pretty much all day, every day, everything, you can describe pretty much anything through physics, but like in this specific application, there's like a really direct lineage to some world-class physics, Berkeley physics that I don't know, led to an insight that basically leads to this more efficacious modality. Can you just break that down a little bit, how that started, where that might take you over time, and
Yeah.
why that's so important, actually?
Yeah, absolutely. And this really, this is sort of speaking to the question of, after we decided that this speed thesis was a real thing and we were going to go after it, why did we develop an inhaler? Um, and so I'll start there with it, basically with the question of, okay, if you want to get medicine into the body essentially instantaneously, how can you actually do that? There are only two ways. One is brute force, stick a needle into somebody's vein with an IV injection and then just directly inject it into the bloodstream. That works incredibly fast, but it can only be used in a clinical setting, so it isn't an actual product that anybody can take with them and use on a regular basis. And so if you're trying to put a therapy in the hands of millions of patients, that's just not a viable option. And so with that off the table, it only leaves you with one other option, which is where the inhaler technology came from, which is that you have to use the pathway we've evolved for breathing. And this is where the aerosol physics element comes in and becomes really interesting. So the pathway that we've evolved to breathe, which is essentially you inhale oxygen air into your peripheral region of the lung. We just sort of colloquially talk about it as the deep lung. You inhale it into the deep lung, and then that diffuses across the alveoli lining of the deep lung and into the bloodstream. That process happens incredibly quickly. And if anybody wants a fun test to do, hold your breath for as long as you can, and then finally breathe when you're no longer able to keep holding your breath, and instantly you feel that relief. That's how fast that process works. And it's evolved that way out of necessity. We need to be able to get oxygen into our bodies, at a moment's notice. And so essentially, if you can hijack that pathway, then you can use it to deliver medicine as quickly as we are able to breathe air. That's a really challenging thing to do, and that's where the physics angle comes in here, because along with evolving the ability to get oxygen in super quickly, we also evolved really good defense mechanisms to keep everything beside oxygen out of that part of the lung, because you don't want to be inhaling, environmental contaminants and getting them into your bloodstream. And so it becomes an interesting physics-based question of how do you create an aerosol drug particle that can basically mimic the gases that we're breathing in around us and use that pathway for delivery into the body. And so the way that we did it is essentially primarily particle sized based. So I think the easiest way to talk about this is to compare it to existing inhalers that are on the market. So people are familiar with like meter dose inhalers, little puffers, dry powder inhalers. These types of products exist. They all create particles that are categorized as large particles, meaning they're 5 microns and bigger. And so what happens when you use those, when you breathe them in, is they get trapped by the central lung. That's where most deposition happens, is in the central area of the lung. That's really our primary defense mechanism in the lung. You get a lot of impaction there, so particles just hit the walls there. You have a really thick mucus layer that's good at trapping things and holding it there, and then not allowing it to get any deeper. And delivering medicine to that area works perfectly fine if you're going to be treating asthma and COPD, because those are conditions that exist in that part of the central lung topically. So you're just sending it right there, it acts topically, and then everybody's happy. But if you want to be able to bypass that part of the lung to get the systemic absorption for something like migraine or pain, then you have to essentially engineer particles to be small enough that they don't get trapped like the bigger particles, they behave like the gases. And so a lot of the development that has gone into our inhaler is how do you create these aerosol microparticles that are smaller than 3 microns, which is really the cutoff for where it's small enough to behave like a gas more so, and it can actually follow the air streams. It just bypasses the central lung and gets that deep lung absorption. And so there's been a lot of sort of engineering and physics modeling of how do you actually create these really small particles, and then also how do you productize that core microparticle technology and package it into a portable inhaler that you can put in the pockets of 1 million people. And so that's really where our sort of physics angle comes in, and why the early days of the company, it was really all engineering and aerosol physics focused. And of course now we have a chemistry team and everything like that, but yeah, the earliest days was actually validating that we could do the physics piece and the engineering piece. That's what we then took into that trial and demonstrated. And now that we have that platform developed, sort of that's an answered question, and now it's about applying it in different therapeutic areas.
Kevin, I know you had some questions too. Do you want to hop in?
Sure. Yeah. Yeah. I'll jump in here. Thank you, David. So, Dalton, maybe you could just talk a little bit for the participants here today about the clinical trial design for the migraine product, and how you're thinking about recruiting endpoints. How do you get to that critical 10-minute window during which the inhaler would presumably need to be utilized in order to see the rapid absorption and onset that this will deliver. So maybe just a little bit about that, because the compressed development time frame is really quite dramatic, and maybe you could talk a little bit about that from a landscape standpoint, where the comparable time frame for a lot of drugs can be easily 6, 7, 10 plus years to accomplish what you're able to do very quickly. So from an investor standpoint, that speed to market is very attractive to us, but that clinical trial obviously will be critical in terms of design, enrollment, endpoints, so on and so forth.
Yeah, absolutely. So for us, the nice thing now is that we've met with the FDA and we've discussed 2 programs, and for each of those programs, the FDA has come back with the same answer and the same pathway. And so what that tells us so far, which is the 505(b)(2) pathway, so it's sort of an expedited approval pathway. And what that tells us so far is that there is a recipe for this approach that we have, and that we're likely going to be able to use that recipe for all the drugs we're developing. And so, the benefit is that we have gone through that for our first program to a certain degree. That makes us better at going through it the second time, and sort of there'll be a nice flywheel effect here where, every time we take a drug through this pathway, we just get better at executing it. And so, perhaps we can even condense our timelines further as we get more and more experienced with this. But there's three general pieces of the path. We get a drug in-house and we test it in the lab. Then we take it into a lung study. In canines, we show that it's safe in the lungs. We've done that with both of our programs that we have now. And then we go into a human PK study as the second aspect, which we've done with the first program. That's the data that I was showing there. And you just validate, the speed with which it works in humans. That's what the FDA wants to see to tick the PK boxes. What does that curve actually look like? And then the last step is that you take it into a study where you're either looking to show some level of safety or efficacy depending on what the FDA is requiring there. And the study design of that, I think, is what you're asking about. And the nice thing about the programs we have right now is that because we're working with drugs that have been approved before, there's largely a template to follow for what that last trial should look like. Right? We can design it based off of the trial of the drug that was previously approved and that showed sufficient efficacy to actually be accepted by the FDA. And so it gives us a really good starting point. We're not trying to create new endpoints that have never been tested or new designs that, have actual clinical design risk in them, which is really nice. And then the way that we, just optimize for our therapies are, we can add things into the trial. So for example, with migraine, we'll largely be using the designs that have already proven to work and be accepted by the FDA. And then we'll add things in like prospective stratification based on how quickly somebody uses the inhaler. Right. So our inhaler is fully software controlled. It actually logs when you use it. And so we'll know every time somebody doses with the inhaler. And so what we can do is with that data between that and patients being observed and when they have the start of their migraine and everything like that, we can know how quickly you use the inhaler after the start of your migraine. And by prospectively stratifying on that, we can show that the faster, assuming our thesis is accurate here, the faster you use the inhaler, the higher the efficacy goes. And that's really nice because once the product is approved and it's out there in the world now, patients and more importantly, physicians who are going to be prescribing the product have something to look at and go, okay, if you use this at the first sign of a migraine, the efficacy is best in class. And so when they're talking to their patients and prescribing it, they're going to be able to give that instruction. And so there's little things that we can add in like that to really optimize the way that people actually use this to get the most out of the product.
Great. Thank you, Dalton.
Yeah.
Um, yeah, just wanted to just tell the audience again, just put your questions into the Q&A or the chat feature. I see a hand up, but just go ahead and put it into the into one of those two, and we'll get to all those questions. Um, Couple of questions here. They're somewhat related, or I'll kind of bind them together. I know you just talked about the FDA process and the development process. Could you maybe articulate um, what that journey is going to look like for you guys, where you're at on that journey? And then I do think there's comparisons to other types of products or modalities that are out there. I see a question around how is it different than the ones that are out there for asthma and acute allergic reactions, um, and apparently limited to small soluble chemicals. So maybe just hit on a couple of those things in terms of just the known path ahead, and then just how others are maybe addressing analogous approaches.
Yeah, sure. So I'll start with the second piece of that. So as for the difference between what we're doing in your standard inhalers, that largely comes back to the particle size piece of this equation, which is that with a standard MDI, DPI nebulizer, you're creating these large particles that are perfectly suited for treating asthma or COPD. But since they're getting deposited in the central lung with that thick biomucus layer, you're not getting fast absorption into the bloodstream. You get minimal bloodstream absorption. And honestly, a lot of that actually comes from you end up clearing the drug out of your lung and then swallowing it later. And so you end up getting some bloodstream absorption from the actual drug that you're swallowing, as well as some minimal through the lung itself. But all of those technologies, they're not designed to get drug into the bloodstream. They're designed to keep drug in the lung, treating the topical site of the disease. And that's very different from what we're doing, where we are creating these microparticles that are smaller and able to get into the deepest region of the lung, where they rapidly diffuse into the bloodstream. And that region that we're targeting, the lining that separates the lung from the bloodstream is called the alveolar epithelium. It's about 100 times thinner than a strand of human hair. So it's essentially one cell thick. And so then that leads to the second piece of that question, which is that it's actually very non-specific in terms of what goes across it. Because that pathway evolved to prevent something from getting there in the first place, it itself doesn't really have any defense mechanisms. And so you're able to get a lot through that alveolar epithelium if you can get it to that deep part of the lung. And so we can deliver small molecules are very easy. And it actually, it's okay if they're either lipophilic or hydrophilic. They both go across that membrane. The difference is how are they getting across? Is it transcellular transport? Are they going through tight junctions? But all small molecules very rapidly diffuse across that membrane. And then you can also use peptides and proteins as well. The primary cutoff is just molecular weight. So once you get to about 10,000 Daltons in molecular weight, you get molecules that are too large to actually effectively go across that membrane. They're too large to go through the tight junctions, and they're too large for transport across an actual cell. And so there is pretty much where you reach your biggest limit. And this has been shown with, so you can do things like insulin, for example. Insulin is absorbed rapidly through the peripheral lung, as a peptide. And so there's a very wide range of drugs you can actually deliver using this method. initial question, David, was the first piece of that was about what?
Well, the want to hit on the FDA process and just what that might look like. There's even another question that's popped up, just like ongoing human clinical trials or where you're at in the journey, if so, where. Just maybe just unpack that because that's one thing that for those who aren't as familiar with biotech and biopharma is there are known gates, of course,
Yeah,
for safety and efficacy, but just there's stuff even before that as you start to qualify where you're at and
yeah.
have initial discussions and all that.
Yeah, yeah. So I'll talk to that with migraine right now since that's our primary area of focus. Um, so if we go back to sort of that three-step framework that I talked about, which is first show that's safe in the lung of an animal, then run a human PK study, and then run your pivotal study that gets you approval. Those are the general three steps. Um, so more tactically for migraine right now, we are we ran our animal lung studies. We showed in those studies that it's safe up to 47 times the human dose, which is a very strong safety multiple. And so basically what that's doing is that's answering the question of, nobody's ever inhaled these drugs before. So when you deliver the drug to the lung, do you see anything unexpected, any toxicities happening in the lung itself? And so, we've demonstrated that you can deliver 47 times the dose we're actually going to deliver to humans, and you don't see any abnormalities or toxicities in the lung. And so that sort of generally checks that box of lung safety at the first level. So then what we do is we take that data, we go and we meet with the FDA, which we've done at this point. We talk about, okay, this is what our data looks like so far. We're going to run one more canine study just to confirm that, and then we're going to go into humans. This is what our first human clinical trial looks like, and it's really all about aligning with the FDA on that. And so, that was a big recent push for us was designing that first study, meeting with the FDA, getting all that clarification at this point, which we have. And so we're at the point right now where, the next big clinical milestone for us is going into this study that we've designed, and it's focused on PK, pharmacokinetics, meaning that what we'll do in that study is we will show how fast our inhaled sumatriptan is actually absorbed and how fast it's absorbed relative to the other treatments that are out there. The FDA allows us to use, it's what we call comparative bioavailability to actually serve as a safety basis for our treatment. So, when you take a drug, anytime you take a drug, you're creating certain concentrations of drug in your bloodstream. And what the FDA allows us to do basically is to run these PK studies and show that, okay, if your total exposure of drug in the body is less than or equal to that of the sumatriptan pill, for example, then you can use that as grounds to say that it's safe because you're not giving more drug to people than what we already know is okay and safe. And so this first PK study for us not only checks the PK box and shows that it works fast, but it also checks that safety box and gives us a large degree of confidence that we're not going to have any safety issues. And it's pretty easy for us to meet that metric because we can actually deliver really low doses of drug. So, for example, sumatriptan, people take pills that are 100 milligram pills, and our inhaler will only need to deliver 2 to 4 milligrams to get that, where we think you get best in class efficacy. And so our exposures are so much lower that we really easily sit within that window, which is quite nice. So that's our next big step for us as a company is running that study and demonstrating that in humans. And then once you have that, the last thing the FDA wants to see is a larger scale study of patients actually using this. And so that's where we'll just have, your classic placebo control design. So we'll give people our inhaler with sumatriptan in it. We'll give people our inhaler with saline in it, and we'll go and have them treat 3 to 5 migraines over a month or two. And then based on that, we'll actually see our efficacy relative to placebo. And that's the data that we'll use to support the approval, but then also to support sort of our post-approval physician adoption statistics. That placebo comparator allows us to stack it up essentially against other drugs that are out there. And so we can use that to support the physician adoption piece and show that, we have the best migraine relief rates of the drugs that are out there. So that's what that pathway looks like at a
Yeah.
high level.
Okay. Yeah, and there's actually a good question in the chat that I'll just read verbatim because it piggybacks off of that physician journey and just what this might look like clinically. So I'll just read this. I'm curious what the patient journey will look like. I assume the device is prescribed by a physician, and then does the patient load their generic medicine into the device themselves? Second, how do you think about the competition of big pharma potentially pushing their own inhalers, especially if standard inhaler peak drug concentrations are good enough during the clinical treatment window? I get migraines too, and totally agree. No medicine ever helped once the migraine took hold. So that's, yeah.
Yeah, gotcha. Okay. Um, yeah, so let's start with the physician adoption patient journey piece of that. So it is going to be a prescription therapy. You're spot on with that. It's actually so it's regulated as a combination product, and so you as the patient, when you get the prescription from your doctor, you're getting everything. You don't have to, separately load any drug into this, which is a difference from typical inhalers that I think is beneficial for us. But this comes preloaded. So this comes preloaded with a month's worth of drug. So once you get it, you're good to go. You have it for your month. It'll give you your 10 doses of triptan that you can use however you like throughout that month. So it's pretty simple from a patient perspective to actually use this thing. There's no, like, if anybody here's ever used a nebulizer before, because it sounds like maybe you have and you know the filling aspect of it, with those, you have to fill them, use them, empty them, clean them. It's sort of a hassle of a process. So we've engineered all of that out of this and made it a device that's pre-filled. You can literally just pick it up and use it whenever you need it. Going back to the actual physician-patient journey piece of it, one of the advantages here I think that we have on the physician adoption side is that when you're working with new drugs, you're always facing the uphill battle of switching patients to a different drug that can have different side effects. And within migraine, sumatriptan is the most widely prescribed drug that is out there. And so you already have majority of patients on sumatriptan. And when it comes to getting people to use our inhaler, we don't have to convince physicians that they should put their patients on a different drug. We just have to convince physicians that they should put their patients on a more effective version of the same drug they're already using, which is a lower hurdle for switching. And so, because of that, I think we're likely to see a faster initial uptick in physician adoption than you might get with a new chemical entity that needs to be proved out a little bit more. And then also on the patient side, you sort of spoke to this yourself. Patients want something that's faster, and it's a very easy story for them to understand. So when you're dealing with new chemical entity drugs, there's not an easily understandable mechanism there for a patient to go like, oh, I really like the way this drug hits this receptor and has this effect. Whereas in our case, a lot of the migraine patients that we've talked to, they're taking sumatriptan, we sort of tell them what we're talking about, and the reaction is, okay, so this is just the sumatriptan that I'm using, but it's going to work in 30 seconds instead of 2 hours. Like, I want that thing. And so I think from both a physician and patient angle, it's a really simple story that they understand and that they want. And you have a lot of patients right now that they're just not willing to use like my wife won't use the injectable drug. When she's already in the throes of a migraine, she's not willing to stab herself with a needle and inject herself. It's not worth it for her. And so you have patients that really want this speed, but there's nothing out there that they're willing to use right now. And I think we can give them that for the first time. Um, so that, that's sort of the way that we're thinking about the market a little bit there in terms of what patients and physicians want. When it comes to the big pharma piece of this equation, I think one thing here is that there we're sort of outside of the wheelhouse of big pharma a little bit. There are some big pharmas, GSK is a good example, that have pretty strong engineering divisions, but most of them don't. A lot of your big pharma companies are not developing complex delivery technologies. They're focused on their drugs and new molecule development. And so it would be a relatively big change of pace for somebody to go ahead and do this. But I do expect that at some point in the future, we will see a change here, right? So let's say we get our migraine drug to market, maybe we bring another drug to market. We, show patients that you don't have to wait 2 hours for your pill to work. You can literally have relief within 1 minute. I think once you show people that this is a reality, there's not going to be any going back after that point, and that you will see the world of acute medicine shift towards this idea of these instant acting inhaled therapies. And so I would expect to see an uptick in competition once we show that this is possible. And that's where then we get to have, some level of fallback on. We have a significant amount of IP that we have filed on this technology and the ability to create small particles, as well as, we have in-house production processes for actually making this happen. And so we're in a pretty strong position owning the hardware, owning the critical supply chain elements, owning intellectual property on this. And so for a big pharma company, if you're developing a new drug and you want to get it into the body instantly, and you have two options of, okay, well, we can either start up our own internal program, which is likely going to take a few years to try to work around Miist's IP and create a competing technology, or we can just partner with Miist and put our drug in their inhaler, and give them a royalty and some milestones based off that, which is really common in the industry. I think that's the more likely path that we're going to see. And so we're starting with our own drugs that we can own the revenue streams of, and then after that we'll expand and we'll entertain some partnerships with pharma, and I think we'll see a lot of good synergies there.
Yeah. The thing that I keep coming back to is that word physics. I mean, it's just such a interesting, because of what you guys have been able to do in the lab, and it just enables just a whole new way of approaching some of these problems.
Yeah.
And you brought a fair amount of that now in-house, which has been interesting to see, because you were pushing your partners pretty hard on what they were going to be able to do, and you were like, you know what, I think we just need to own this strategically. So that's been really cool to see.
Yeah.
So yeah. So
Yeah, there's a really small number of people in the world who are expert in this specific niche, which
yeah,
is advantageous for us in a lot of ways. It means that if you are the best in the world with aerosol physics or aerosol drug delivery, there's not a, there's not a lot of places you can go to do the best work. And so, here has been the home for a lot of those people, which is really nice. And so it's great for us, and it also means that there's just not a lot of you can't create 30 of these companies. There just isn't enough talent to actually even do that.
yeah. And it's one of those, or it seems, having been on this journey with you, it seems like it's one of those situations where you're like, oh, nope, I didn't realize that nobody was going to be able to maybe help us with that. So let's just build it ourselves. And then that creates more IP, it creates more defensibility. What maybe wasn't the initial path, but it's like, oh, and we were able to do that. Okay, great. We were able to build that team with that diverse skill set. And so it's creating the moat for you along the way.
Absolutely. Yeah.
So yeah. Well, I want to make sure I know we're now just even a little bit past the top of the hour. I know Jonathan had a very interesting question. I don't think we have time to address it now, but just to, end this, on time. But I did put your email address into the chat. So Jonathan, others, if you have questions for Dalton, or just want to connect with him directly, feel free to email him. I didn't put your LinkedIn profile, but he's also on LinkedIn if you want to connect with him in that way. Um, but yeah, thanks so much for joining, and we're really pumped about what you're building, and think it's very exciting, and obviously business opportunity for investors, but also as I always like to say, that's why we started this firm, was to help founders solve big problems, and then of course investor returns are going to come, but just you're going after some really important stuff, starting with migraine, and there's a bunch of other things you touched on that basically where speed is the number one, obviously efficacy as well, but just that speed variable is just so important. So thanks for carving out some time. I know you're busy, so just yeah, thanks again for carving out some time, and again, thanks for everyone who attended as well. Really appreciate you joining us this morning.
Great,
Yeah.
great work, Dalton. Thank you. Good to see you.
Thank you. And thank you guys for having me.
Yeah, thank you. Take care.
Bye, everybody.
Bye.
Bye.